So, Laura, good to see you.
Hi. Yes, I just managed to. It just everything got minimized, so I couldn't see anything anymore. Hi, nice to see you, Kenny. After I think it's been 3 years now. Just wanted to thank you first for funding through the redistributed manufacturing in healthcare network this project, and for the invitation for presenting here today.
No problems. You go. All yours.
Thank you. So I'm going to talk about this project that was funded, and it's all about ultrafast 3D printing for in deployed situations like the battlefield, for example. In this case, in this project, we're focusing in the wound fillings for immediate stabilization of soft tissues, but this kind of printing can be used for many other different applications. So before I start talking about the project, I just want to present the team. Infant, who is leading the project with me, and all the other collaborators from Nottingham, the researchers, the collaborators from Lawrence Livermore, and the collaborators from the Scar Free Foundation, and our advisor from the STL, Abigail Spear. So to talk a bit about the background of this project, for in the battlefield, when we were talking with our collaborators and our advisors, we realized that the main treatment that they do to stabilize wounds in the battlefield is a negative pressure wound therapy. How does this work? Basically, they apply negative pressure on the wound. The filling material, it can be a foam or a medical gauze, that goes in here, and then they apply the pressure. So and with that negative pressure, all the liquid gets drained. But this also it increases the cell migration and proliferation to start the healing of the wounds. But what is the problem, or the problems with this, is that sometimes like the foam doesn't perfectly fit on the wound, so there is problems with the sealing with the machine that applies the negative pressure. And as well, after the treatment, when the patient gets taken to proper treatment into the hospital, when they remove these fillings, sometimes it disturbs the healing of the wound, because the tissue started migrating into the wound. So the idea here is to create a personalized wound filling, and also with a material that can be disintegrated without having to physically remove it. So just add something to trigger the dissolution of the material to be able to remove it after a few days. So in this project, we're combining the negative pressure wound therapy with computer axial lithography or volumetric 3D printing, which is a type of printing which is very fast, and I'm going to talk about it in a minute. So just to give an idea of the project, is you have the idea is to use a handheld scanner to scan the wound, and then use that data to create the model for the filling, printed with the volumetric system. And also the idea is to have the inks already in vials that are ready to use. So the vial doesn't have to be opened, you don't get any contamination or anything, you have it prepared, put in the printer, print, and then get the filling ready to put for the wound therapy. So this is let me just start the video. This is the volumetric system that it can be deployed, and you will see the real system in a minute. But this system was developed in Lawrence Livermore National Laboratory. And how does this work? So instead of working like all the other additive manufacturing technologies that they work basically constructing something layer by layer, in this system, the material, the model gets printed all at once. The system uses UV polymerizable resin. And in Lawrence Livermore, they develop a software basically that instead of slicing a 3D model layer by layer, it slices the 3D model in different angles, like the CT scanner. And then these different slices get projected towards a rotatory vial in order to polymerize the entire construct. because the software they develop, basically it controls the dosing of the UV for each image. And that's why the entire construct gets printed all at once. What is the advantage of this? This entire procedure happens within 30 to 50 seconds. So it's very, very fast. And that's the advantage of the printer. So I'm just going to check. So in collaboration with Lawrence Livermore, we built one of these printers in Nottingham and started optimizing the printing parameters with a traditional resin that they were using before there, which is a PEGDAG glycerol-based type of resin. What are the disadvantages of the printer? Well, the resolution is not great. You get 300 microns, but for some, like any type of 3D printing or AM systems, you have advantages and disadvantages. You just select the right type of printer for your application. So in this case, that we want something really fast, but we don't want the resolution to be great, this type of printing works perfect. All the materials had to be photocurable, so you can use acrylate resins, thiolate resins, hydrogels that can be UV polymerized. And at the moment, with the system we got, the maximum size of structures we can get is 5 centimeters tall and 2 centimeters like in length. But we are working towards being able to scale up this and make bigger constructs as well. But this is a very new system, so we're still in development. As part of this project, we also modified the setup. Basically, on the left is the setup from that we obtained from Lawrence Livermore. As you can see, it's very deployable. It's very small. Like the distance, just to get an idea of the size, the distance between the projector and the rotatory stage is around 40 centimeters. So everything it's very small, very compact. And the way we change it is like for them, you have to put the vial on the rotatory stage and basically screw it in. So what happens is that you have to open your vial to be able to put it on the stage. But we adapted it. So because we want to print everything clean, we don't want to be opening the vials because you can get some contamination in there. So we adapted to have the stage at the bottom. So it still rotates, but you just get your vial and put it on top and it will rotate. You don't have to open anything as well. So, and as well as part of the project, we developed a formulation for this specific wound therapy because this technology is so new. There are not many formulations for printing with it because the viscosity has to be extremely high. Why does it have to be so high? Because as the material starts appearing, between those 2 to 3 seconds where it's already being constructed, the material can sink. So we need a very viscous material so the construct doesn't start sinking when it's being printed in those 2-3 seconds. So we developed this formulation. We look into use PEG-based formulations, glycerol-based formulations, many of the different materials, but they were not quite right. They didn't have the right viscosity, or the reactivity is not fast enough for this type of printing. So this is the formulation that we selected at the end. We're using acrylamide as the main reactive material. We're using a triggerable cross-linker. And what do I mean by triggerable? That we can use chelator agents to trigger it after it's polymerized and to trigger the dissolution of the construct. We also have just an alginate as the viscosity enhancer, and of course the solvent water. Then we want to combine either antiseptics or painkillers or growth factors into the formulation to create this tough, stretchable, and triggerable drug-releasing formulation. So in this, we've been working only 3 months on this project. So these are the initial results where the formulation, we're looking at different concentrations of the different parts of the formulation in order to get the viscosity we needed, in order to get the reactivity time we needed as well. So we tested everything, and we have some preliminary printing where we can actually cross-link this material using the printer. So this is just how it looks. You have the vial in there, and then just the light starts flashing as the vial rotates, and that's it. And then the construct gets printed. So what is the idea after we printed it? Then now we're working into the in vitro setup. So we're going to have a cast 3D shape with the shaped on the wound shape that we're going to put our printed construct in there and then apply the vacuum, the pressure. So what's next, or what the summary and the next step? So we adapted the cal system for printing without the risk of contamination, and we also developed this biocompatible, very reactable and stretchable hydrogel for printing the wound fillings. In the next few months of the project, we're going to work into printing porous wound fillings, because the mesh has to be very porous, so the liquid, any fluids that are on the wound can travel through the filling and get extracted. We also need to test the in vitro setup, and of course do some in vitro raw release testing. Unfortunately, I am leaving academia. Actually, my last Friday was my last Friday, and I'm working towards industry, working in industry. So Infen, who has been leading the project with me, is going to continue with this project for the last months. So I just very quickly want to introduce this, because I think it's very related to the redistributed manufacturing healthcare network, to where they were talking about yesterday, about creating biologics or creating everything in deploy situations. So this is the new company where I'm going to be working, which is Biologic Technologies, and are developing this biocomputer platform. Basically, it's a miniaturized type of bioreactor that can be used for manufacturing either antibodies, gene therapies. It can be used for manufacturing also stem cell therapies. And the reason why it's so versatile is because the components are all 3D printed, so it can be fast adapted to the different applications. It has all fluidic control, electronic control, thermal, optics, pneumatic, and biologics. So it's all miniaturized, so it can be used very easily in different locations, and everything is fully enclosed. So it's just like, I think it's very related to the redistributed manufacturing and healthcare network. So I just wanted to introduce that, and just want to say thank you. And if you have any questions, also you can contact me here, my email, or if you have any questions about the other project, you can contact Infeng as well. And that's it. Thank you very much.
No problems. Thank you, Laura. And yes, and hope everything goes very well in the new place.
Thank you.
That's all right. Fourth session, we have 4 presentations, all essentially related to design processes and around kind of medical devices and healthcare applications. I'm for this, and then we have a final discussion, and then we'll get to the end. And then the first of those presentations is Jari Pilari from Taika 3D. He's going to talk about design automation. And so if I stop sharing, Jari, you should now be able to, hopefully.